So I mentioned earlier that the capturing of this fluorescence is really at the heart of a real-time PCR experiment, and that is very true. And to simplify a very, very large area, I have broken up the fluorescent labeling systems into two types. The first type being cybergreen, and the second type being the fluorescent Quencher probes. So if we look at cybergreen, you may have heard of it before. It is a dye which binds non-specifically to double-stranded DNA, generating a DNA dye complex which emits green light, and it is this green light that is recorded by the real-time PCR instrument. So as you will see on my homemade diagram here, when cybergreen is on the first half of the diagram, I'm showing you the very beginning of a PCR reaction, but it could also be a negative control if the primers, as indicated by the arrows, were emitted. When cybergreen is in solution and not bound to double-stranded DNA, it emits a very, very low fluorescence. However, on the right-hand side of the diagram, we can see that as amplification proceeds, the primers are extending the template, and we get the formation of newly synthesized double-stranded DNA. And then cybergreen can bind to this and emit a fluorescence. So it's a very neat and very powerful technique, but it has one major drawback, and that is that because cybergreen binds non-specifically to double-stranded DNA, you risk the detection of off-target amplicons. But there are some tools that you can use to try and circumvent this, and one such tool is running a post-run melting curve, and this I will come back to later in the talk. The second type of labeling system encompasses the fluorescent Quencher probes. So in the first part of the diagram, I'm showing you at the beginning of a PCR reaction where we have our DNA template, single-stranded, forward and reverse primers, and then we have the probe. And the probe is something which is comprised of three parts. The probe itself is actually has a specific region which is able to identify a complementary base pair to your target sequence. And attached on the 5' end, we have a fluorophore which is covalently attached, while on the 3' end we have a Quencher. And when the probe is intact, so when the fluorophore and the Quencher are in close proximity to each other, the Quencher prevents the fluorophore from emitting any fluorescence. And then in the second box, we have PCR amplification is underway. So during extension, the polymerase is being brought closer to the probe. And then in the third box, we see where the polymerase has met the probe, and the inherent 5' to 3' endonuclease activity of the polymerase actually cleaves the probe. It needs to cleave the probe to continue extending along the template. And when it cleaves the probe, the fluorophore becomes released. And in that way, it cannot be quenched any longer, and it emits a fluorescent signal, and we can quantify this. And again, the quantification of the fluorescence is proportional to the amount of amplification taking place, which is a representation of how much template we have.
To view the full video of this and all of our other webinars for bioscientists at the bench, please visit bitesizebio.com/webinars.